So welcome to the 2 o'clock session of the tomato meeting, sometimes called nap time. So we'll kind of try to avoid that. So anyway, my topic today is to talk about several new biopesticides that have come onto the market very recently, both from the company I work for, as well as others in the industry, and talk a little bit about their uses and applications. I do work for a publicly traded company on the NASDAQ exchange, so our legal department would always ask me to put this up there, and remember, as Ken said, there will be a test. So now this is just a reminder that I may make comments about future product registrations and introductions, and of course those cannot be guaranteed. So you don't go to the Mid-Atlantic show for stock tips. So I'm going to be talking about biopesticides, and I thought it'd be worth spending a moment to just sort of define the topic, because we use the word bio a lot. We talk about biofertilizers, biostimulants, and biopesticides. Biopesticides are materials that make a crop protection claim, whereas biostimulants are typically for crop enhancement. They might be to mitigate stress, but if you make a crop protection claim, you're a biopesticide, and therefore you're regulated by EPA within their BPPD division. Biostimulants cannot make any crop protection claims. Sometimes they push that envelope a little bit, and they're regulated by the states, usually under their state fertilizer laws. So in the United States, with EPA, we have two different biopesticide categories from a regulatory point of view. We have microbials and we have biochemicals. Microbials are, of course, microbes. They're either based on fungi, bacteria, viruses, or protozoa. Biochemicals are typically plant extracts, such as azadirachtin or renutria extract, pheromones used for mating disruption, soaps and fatty acids. And of course, biopesticides have a very long record of safe use, 70 years now, and typically are more timely to get registered and less expensive. And it's kind of worth mentioning that because I'm going to talk about 4 different products today that have come onto the marketplace essentially within the last year. And we kind of live in an era where there's not that many new product introductions. And part of the reason, of course, is that in the conventional side of crop protection, the number of companies have become fewer through different mergers and acquisitions. But a lot of it has to do with the cost of registering a new active ingredient. So on the chemical side of the business, it takes roughly 10 years and $300 million to bring a new molecule to market. That's a worldwide launch. And typically, of course, as everybody knows who grows fruits and vegetables, they're targeting some of the big acre crops, and then hopefully they also get some minor uses registered. Whereas biopesticides, you can typically discover a new active, get it through the development process, say, for example, 5 years at a cost of $4 to $8 million. Now that's a US launch. If you want to launch worldwide and get it into Europe and Asia and Africa and South America, you're going to spend another $10 to $20 million. But it's a lot less expensive than the conventional side, and it's one reason why the biopesticide segment of the market, which is still very, very small, but it's growing at about 3 times the rate as the conventional side of crop protection. So this is a slide that I've kind of used in these sorts of presentations over the years. It kind of shows some of the products. It's not all inclusive, but basically on the left are microbial-based materials, and on the right are the biochemical examples. The one on the left is obviously a bigger list. It's grown a lot in recent years. One area that's grown a lot in the last decade or more are fungicides based on different bacillus isolates, and we'll talk about a couple of those today. A couple of things that I think are worth noting in general. These 3 products here, if you grow corn, soybeans, or cotton, you probably have Votivo or Clariva, or another one called BioST as a seed treatment. So biological seed treatments have become very common, and they're being used on tens of millions of acres. And the last one here is a biofumigant that I'll also mention in my presentation. So I think it's worth noting that when we talk about tomatoes in the Mid-Atlantic, we're really talking about 3 very different crops. We have field-grown processing tomatoes, we have field-grown fresh market tomatoes, and then we also have fresh market tomatoes that are grown in some sort of a protected environment, whether it's under glass, plastic, and a hoop house sort of thing. They have very different agronomic characteristics, they're different varieties, and they have different pest and disease complexes. So the products that I talk about, some may have use in one of these 3 segments and not in the others, where others will be useful potentially in all 3 segments. So I'll talk a little bit about nematodes. It's one, I've been coming to this meeting for a long time. We tend to not hear much about nematode management. Certainly in Pennsylvania soils, we have pretty heavy soils. They're typically not a problem. As you get closer to, say, the Chesapeake Bay and in river bottoms where there's a little bit more sand, you certainly might have some nematode problems. Nematodes are not easy to manage, and they're not easy to develop products for. They're nearly invisible. They're microscopic roundworms. The damage is not always visible, especially for free-living ectoparasites such as lesion nematodes, where you get sort of a decline in crop health, but you don't really see any other obvious symptoms. They're also difficult to sample because of their very clump distribution, so it takes a lot of soil samples to get an accurate measurement, and all those soil samples cost money. Now, in some cases, such as the ones we tend to get in tomatoes, root knot nematodes, the damage is pretty evident. You can simply pull up a plant and you can see it on the root. Other common species would include reniform nematodes, cyst nematodes, which are also quite evident, and spiral nematodes. And apparently nematologists are pretty simple folks. They look at the nematode, decide what the shape it is, and that's how they name it. We're a little more creative as entomologists. It is important to note, though, for nematode management, that we're somewhat our own worst enemy. Nematodes are small little fellows and little gals. They don't walk very far from me to the end of that stage. That's about how far a nematode can spread in 1 year. So if you have a nematode-infested field, make sure that when you have machinery in that field and you take it out of there, that you get the dirt off of it. Because for the most part, nematodes spread by human activity. So the first product I'm going to mention is from the company I work for, Marone Bioinnovations. It's called Magistine. What it is, if you pick up a jug, it contains Burkholderia rhinogenesis, a unique strain that was discovered internally. It's a liquid formulation that contains heat-killed cells and spent fermentation media. So that means it's a metabolite-based product. The active molecules are in this fermentation media. And what it does to the nematodes, it's fairly broad spectrum dependent on the species. It can reduce egg mass formation. It can prevent molting from juvenile to adult. And it's worth noting that nematodes and insects are the two animal groups that have a true molting stage to different physical characteristics, if you will. They actually share a common ancestor a few 100 million years ago. And at the right concentration, the product will cause direct mortality in the free-living stages that are called Tues. So a few other characteristics about the formulation itself. It's fairly stable, has a shelf life in excess of 2 years. It doesn't have to be refrigerated. It has good mixing characteristics. As many pesticides are, it's exempt from residue tolerances. Has a short 4-hour re-hour entry, 0-day pre-harvest, and it's an organic listed type product. So the example is a sort of level of control that you might expect. So this is a study down in Florida for root knot nematode on fresh market staked tomatoes. Typically they use a fumigant before production. These are grown on plastic. Metam sodium would be a standard. We're looking at a galling index, so the lower the number, the better. But once you use metam sodium, you're not going to completely sterilize the field. Only from the depth you go up, the nematodes can come back in the season, so you traditionally don't have a lot of in-season options. So that's our standard, and then in comparison we have the Magistine at 2 different rates, 2 and 4 gallons per acre. Through drip irrigation, the A here means that it was first applied shortly after transplant. The B means a second application about 28 days later. So we can see that the performance is very comparable to the fumigant standard. Not much difference between 2 and 4 gallons, a little bit of an improvement for the root rating, not much difference in yield. So from an economics point of view, the 2-gallon rate applied twice is sufficient, and that's the maximum label rate of the product. So some best use recommendations for this type of a product is you're going to start with 2 gallons per acre, either in the transplant water or more likely through drip irrigation shortly after transplanting. So it's a product that it can be applied in furrow, but probably the most efficient means would be through drip irrigation. And then you're going to have another application 3 to 4 weeks later, potentially one more after that if the nematode pressure is sufficient and the economics warrant it, but that's going to be at a 1 to 2 gallon rate. The key thing about this product, it's all about dilution. You can over dilute the product. So it's not the sort of product that you can go through center pivot irrigation very effectively. You have to be very careful about water volume. So the most efficient way is through drip irrigation towards the end of an irrigation event where you maintain a critical concentration and you get a high level of efficacy. A soil adjuvant to enhance soil penetration can also improve product performance. So I'm going to switch gears a little bit now and talk about two fungicides. They're both based on bacillus isolates, and they're very, very different in terms of their mode of action. So the first one is called Stargus. It's a new foliar type fungicide. Also has some soil applications. I'm going to talk mostly foliar here. It's for control of some of the more difficult species of diseases, white molds, downy mildews, Botrytis alternaria, Phytophthora, and it does also look like it has activity on bacterial diseases. So it's a very different product than our Regalia fungicide. It does also have some soil applications that we've been looking at on potatoes for Phytophthora and Rhizoctonia. So from a product definition point of view, what is it? It's a liquid formulation. It's a 1 to 4 quart per acre rate, depending on the crop and the disease. The active ingredient is Bacillus amyloliquefaciens, strain F727. It's fairly broad spectrum due to the peptides it produces during fermentation, but we are seeing a fairly significant level of SAR activity, and that's a systemically acquired resistance that causes the treated plant to produce plant defensive compounds. So this is sort of a recent part of the discovery of the SAR. We kind of need to quantify it as to what level of contribution it's making. We think the predominant activity is still these peptides. So the FRAC code, which is the Fungicide Resistance Action Committee code, is still pending. Exempt from residue tolerances, armory listed, broad tank mix compatibility, the usual 4-hour reentry and 0 data harvest. So it's interesting to note that there are a number of bacillus-based fungicides on the market that are basically peptide-based or lipopeptide and SAR active. So the question, I think, a very obvious one is, well, what's the difference? And if you look at the fermentation supernatant from these different products, and this is Stargus at the top compared to 2 other commercial products, you see that you get different liquid chromatography sort of printouts, if you will. So it shows that although these products may actually be in the same genus and species, they're different isolates, they produce different biochemicals during fermentation, and that's why you see them with different labels for different diseases and so forth. Labeled crops, it's fairly broadly labeled as far as crops, but very specific in terms of its target diseases. So it is labeled on fruit and vegetables, which of course includes tomatoes, primary targets being Alternaria and Phytophthora. Kind of an interesting slide. This shows kind of a significant difference between Stargus and our Regalia product, whereas Regalia has no direct activity on diseased organisms. This is what we call an in vitro study in Petri dishes, where you inoculate it with your diseased organism. This is Phytophthora capsicii, and this is treated on the surface with a dilution of Stargus, which is coded in BI 110. You can see that it inhibits the growth of the pathogen until you get to a very, very dilute rate here, and this is the chemical control. So it does have direct activity on the diseased organism to prevent its growth. Just one data slide. This is actually a potato trial as opposed to tomatoes, but it is early blight and late blight. This is basically Stargus at not quite 2 quarts in the middle here, and I think my little pointer here is kind of losing its juice, and then 4 quart, which is the maximum rate. See a pretty steep dose response, but a pretty good level of activity at that higher rate. So the next product I want to talk about is also bacillus-based, but it's a very different product. It's a very different mode of action. It's called LifeGuard WG. It's from Certis USAID. We have a couple of their folks in the audience, so if any questions come up, I'll be able to direct it that way. But it's a very interesting product in that it's based on Bacillus mycoides isolate J, BMJ for short, but it also does not have any direct activity on the pathogens. So it's a plant defense activator. The FRAC code is P6. So it's the 6th member of that group, 4 of which are available in the US. Quite similar to Actigard, asabenzolar, but it definitely controls different diseases. For example, I've seen it in grape trials on downy mildew, and it's very impressive. Again, it's the SAR type activity. It's been in kind of research for quite some time. It started at Montana State, Barry Jacobson, and it's shown some very good plant safety aspects. Labeled uses, again, pretty broad in terms of crops and diseases, also labeled on fruit and vegetables. Basically, the emphasis is fungi, oomycetes, and bacterial diseases. And it also has on the label for control of potato virus Y on seed potato. I don't know much about that, but I can't say I've ever seen that on a label before. One data slide from them on tomatoes. This is in North Carolina, kind of a mix of early blight and late blight that came into the trial. And it's a pretty good example of how biofungicides in general should typically be used. They do have the product standalone here compared to a chemical standard, which is a rotation of various chemistries, as well as a rotation program where they took out every other chemical, replaced it with a lifeguard, got statistically equal disease control. The same thing with yield, a slight numerical bump. So it's a pretty good example of how most manufacturers recommend that these products be used. So when we think about biofungicides in general, whether it be an SAR type activity or a direct activity on the disease, it's always important to remember these are usually preventative products. There's a lot of discussion on that this morning during the bacterial discussion. You know, if you have a lot of disease out there, you've probably waited a little bit too long. For products that function through an SAR mode of action, it is important to apply to healthy, actively growing plants and not stressed plants. They don't really have the wherewithal to respond properly if they're stressed. One thing to note on the label, make sure for the lifeguard product, you read the label for proper dilution for the proper carrier volume, because both dilution and the actual amount of product per acre are important. And finally, for all of these new products that I might mention today, very seldom does anything get applied alone. We know that things go in the tank, mix it. All the manufacturers, we try to do our best to look at commonly tank mixed materials. We can't test everything or the combinations, so make sure you do a jar test to look for compatibility before you go out with something you haven't looked at before. We don't want any of this. You know? That is not good. That's bad. That looks like a milkshake. Yeah, it's pretty bad. That's a real photo. I won't tell what it is, but you really don't want to have to clean something like that up. That could be pretty hazardous. You also don't want to be the chemical rep who has a product in that and gets a phone call either. So remember your mixing order. You know, you always start with any water conditioners if you're adjusting pH. Biopesticides usually like a pH roughly around neutral. There's a few exceptions, but by and large, that's pretty safe. Start with your dry materials, your WPs, WDGs, and so forth. You know, then you go to your liquids, microemulsions, finally emulsifiable concentrates, and then finally crop oils and adjuvants. Don't do any hot mixes of liquid materials. Don't pre-mix two liquid products together before you put them in the tank. People do actually do that. I didn't believe it till I saw it. It's just a bad idea. Wettable powders and dry flowables, if they're a little bit difficult to mix, you can make a slurry in water beforehand and then pour it in the tank. Sometimes that'll get things in the tank a little better. So the final product I want to mention is quite different than what I've talked about so far. This is called Ennoble. You wouldn't believe how long it took us to get that crop growing in the shape of that shark. It took a long time, especially with the floating leaves. But so Ennoble is a fumigant, and it's a biofumigant. Fumigants in general have typically been quite toxic materials that require specialized handling. Of course, a lot of them are not as available as they might have been years ago. This is a discovery that originated also at Montana State, interestingly enough, from Dr. Gary Strobel. It's based on a strain of Muscador albus, which is a fungus. It's an endophytic fungus, so it has to have another organism to grow on. This is a little bit interesting. So it is an EPA-registered product, but it's not yet commercially available. It's a Marone product, so we're working on fine-tuning the use rate, the application methods, as well as scaling up the production methodology, because it's very different. Instead of fermenting this in a big tank like you do beer, it's actually grown on grain. So it's kind of like making mushroom spawn. So we have a use rate, we're talking 100, maybe as many as 500 pounds per acre, which sounds like a lot of material, but it's really not that much. It comes to it's almost like a soil amendment. So the way this works is you have this material that's been grown on grain, it's kept in a refrigerator until shortly before use. You incorporate it in the soil, it grows, it gives off a bunch of gases in the soil, and that's your fumigation. So it's a pre-plant fumigant, and this is just kind of an example of putting it down right during the same time you're forming your bed, putting in your irrigation tape, put it in that top 5 or 6 inches of soil. You don't necessarily have to have plastic on top. and you get this sort of activity like you would with a fumigant. The exception being that this material does not control weeds. It'll control nematodes, a number of soil-borne diseases, and some soil insects. Just an example of a lab experiment. So here we have the ennoble organism, muscador, growing on this side of a Petri plate. There's a little drop of water that has living nematodes. The gas it gives off basically, crosses a barrier in the plate, kills the nematodes. Very similar, another in vitro test, this being the fusarium colony. And here we have the muscador growing on one side of the Petri plate. Again, the gas goes to the other side and prevents the fusarium from growing. So when I first thought about whether or not to include this product in the presentation, I'm like, well, this is not the sort of thing you're probably going to use in field-grown processing tomato. It's going to be too expensive. Possibly not field market, but for hoop house production, where you have soil diseases in your hoop house, this could really be a very, very good use for this product. So I think sort of in general, it's interesting that, here's 4 new products that have come on the market at a time where relatively few conventional materials are becoming available. The rate of biopesticide research at the university level and within private industry is still very strong. We see this consolidation of the major crop protection companies, which is somewhat limiting their discovery operations. So I think we're going to continue to see these innovative products. I think the quality of products is getting better. They're getting more active on some of the more difficult to control diseases, such as Phytophthora and Alternaria and such. Biopesticides offer growers a lot of flexibility. You typically don't have a limit on how many times they can be applied. They're very compatible in the tank. They can help you stretch supplies of other materials, maybe like chlorthalonil or something that might be a little bit short this year. They offer good short reentry intervals, especially when you have to go in and harvest that crop multiple times. They're typically tolerance exempt, and they're extraordinarily IPM compatible materials. So, thank you.
Okay. We have some time for questions. We're running, we got 7 minutes. Do you have any jokes, Tim?
I didn't bring any fish pictures this time. I should have.
So,
Yes,
any questions for Tim? Yeah, Art.
How about that Magistine is by any chance rated for potatoes as well?
Magistine is labeled for potatoes, yes. In-furrow is the best application method then, and then you could also do it during hilling too. Yep.
Okay.
They're
More questions? Yes.
red.
Go ahead.
Red shirt.
So with the use of this for blight for tomatoes, do you find that growers are using this in combination with other traditional fungicides or different modes of action, or solely this?
Say it again. Sorry.
Okay. Are you finding that with the controlling blight for tomatoes that users are just using this product, or are they mixing it with different modes of action of traditional fungicide?
Well, for the Stargus product, we're actually just launching it this year. We literally got registration in late October. We'll have our state registrations before the crop year begins, across the country. So we actually haven't had growers using it yet for blight, but we would be recommending it in a program with conventional chemicals unless they're organic. Yeah.
Okay, hang on. Coming with the mic. Since we're taping this, we'll hear the question.
Yeah, you mentioned doing the jar test to test like a tank mix compatibility, which is important, so it doesn't clog up your sprayer. But how about combinations which could sort of deactivate the biopesticide?
So the materials that I mentioned here, each one's a little bit unique. So Magistine's a dead product, so in terms of tank mix compatibility, you're not going to kill it. It'd be very unlikely that you would sort of inactivate it. The bacillus-based materials, I think, historically have shown themselves to be quite rigorous in tank mixes. There's really very few things that cannot be tank mixed with. I think the oxidate type products might be a little bit of an exception, might have a little bit of a negative effect. The Lifeguard should be very tank mix compatible because it is viable spores. But I'm actually, Greg, you mind just? Is there any issues in terms of maintaining spore viability?
Yeah, so with the Bacillus products in general, and we have a similar product to Stargus, Lifeguard in particular looks good with coppers, except for some of the liquid coppers. You don't want to mix any of them with like streptomycin or one of those products. So if you're in like apples and you're working in that, if you're looking at, because you guys don't do a lot of strep in tomatoes these days, do you? Just in the greenhouse, yeah. But you don't want to put them with antibiotics, because antibiotics kill bacillus like crazy. So yeah. So some copper issues and some liquid badge. Lifeguard, no. I have to go back and look. We just finished all that testing. I can tell you can't put it with Quavo, but you can put it with Coside. That I know for sure. Right?
I would mention
they're,
that
they're sort of a lot of those are one-offs. Ask us, and we'll get you the information.
Yeah.
All right. So I'd have to go look it up for you.
They call us with what copper product you're using.
Mostly not, but yes.
How's that for an answer? It just depends. It just depends.
Okay. We have time for one or two more. Tim, you have another one? Oh, no.
no,
Yes, right here.
Does Immobile have any action against Rhizoctonia?
which one did you ask about, Enoble or?
Immobile, yeah, the biofungicide.
We
you
haven't tested it specifically on Rhizoctonia, but I would fully expect it would have activity on Rhizoc.
Then eventually,
It's pretty broad spectrum because it is a fumigant.
Yeah, so you could eventually use it for black root rot, like on strawberries and stuff.
Blackroot, strawberry, it's interesting. Enoble, originally our objective for developing that product is for strawberry production. Yeah.
Good.
Yeah, and it's really it's really a California organic strawberry product. Initially, I mean, that's kind of like, when you do your market research and decide, is it worth developing? That's its primary home base, if you will, and then you start looking for other uses, so yeah.
Okay. Thank you very much. Let's give Tim a hand.